you for coming, and hopefully you can hear me fine and see the slides. Okay, great. So yeah, my topic today is investigating bilinear trends in protein structure evolution. Emphasis on investigating. This is very much work in progress, and yeah, I really look forward to your questions and input. And I think there's lots of potential to explore this area over the next little while. So yeah, this is a bit more work in progress than I had hoped, but there's some initial results to share with you. So the talk has two parts. Firstly, my recent work on long-term trends in protein structure evolution, and secondly, going back to some of my previous work thinking around gene origins, which kind of fed into why I started on the current project. So a question to start us off is, are there laws of protein evolution? It's reasonably well known that there are obviously laws in physics, and for instance, the evolution of stars obey fairly reliable, predictable laws that we can understand, and we can work out things like how old a star is based on its properties. Kind of commenting on this, the Cambridge astronomer Arthur Eddington said it's difficult to resist the impression that the evolution of the stellar universe proceeds at a slow, majestic pace. And the question for today is, does the evolution of the protein universe also proceed at a slow, majestic pace? Or is it kind of entirely contingent or ad hoc or historical or environment dependent, which would, I think, be the mainstream view of protein evolution. I want to see if recent data might push back against that a little bit. And I'm curious what you all think about it. So there's some previous work on this idea that there are long-term trends. I won't talk that much about that. I'll just note that this isn't the first work on that topic. So Joanna Mazel's group has published a few papers relating to this theme, where she showed young genes tend to be high in intrinsic structural disorder, and they seem to have more hydrophobic clustering as well, whereas very old genes seem to have decreased in ISD and to be less clustered in the hydrophobic residues. Thinking about this work, I thought that maybe one of the interesting things about it that might be a confounder is that the work is focused on eukaryotic groups, particularly plants and animals. And these have differed greatly in the environment and that they've been subject to, and also their complexity and the cell types that they have. And when you do a comparison of young versus old genes in this context, the very old genes will be prokaryotic in origin, whereas the young genes are eukaryotic in origin, presumably. So I wonder if that's a potential issue, and that you're not really comparing like with like. So I decided that I'd like to try and look at potential long-term trends, focusing just on prokaryotic genomes. Very initial look at what you tend to see if you try and group proteins into very broad kind of age classes. So those proteins which seem to be roughly restricted to the species or the genus at the top tend to be very rich in alpha helices. And this was my kind of initial observation that got me thinking about exploring this more, whereas the very oldest genes that are found across both bacteria and archaea have accumulated, it seems, a lot more beta sheets, to the point where they're almost kind of getting towards 50-50 ratio potentially of those, whereas in the young genes, that's very uncommon to have that kind of ratio. It's usually predominantly alpha helices. So my question as I started this project was, is there kind of a slow gradation, or do genes very quickly acquire what look like these kind of older properties of having accumulated more beta sheets? And the point of this diagram is just that you see roughly the same kind of trend, whichever species you look at. So if you look at all the proteins in Pseudomonas aeruginosa and try and cluster them very broadly into age classes, you see something like this, and etc. across different bacteria, also in archaea. The trend seems to be fairly universal, but this is not very quantitative at this point. It's just quite qualitative. And just looking at this diagram, there's kind of an open question. Is there really much difference between those protein domains, which seem to have arisen at within the order or the class, versus those that arose much earlier? So many kind of open questions just from this first look. And my initial search was just across a few species. There was a collection of domains that had been extracted from some of the AlphaFold models. And basically you see consistently the same thing, whichever species you looked at, in terms of just looking at very young versus very old. But in order to try and have a more fine-grained look at it, I want to try and divide things into phylostrata or age groups a bit more narrowly defined. Phylostratigraphy is the task of trying to divide, for instance, protein coding genes or protein domains into these age classes or phylostratigraphic classes based on their inferred age, or the largest taxonomic group which shares the gene broadly. And it's by analogy to strata in geology that in geology, roughly, the layers near the top of the geological column are associated with younger age, whereas the very oldest layers tend to be towards the bottom of the geological column. So this idea that you can classify according to age by analogy has been extended to genomes. You try and classify genes according to how widely they're spread taxonomically. So if something arose just very recently, it'd be very surprising if it was spread more widely than, say, a species or a genus. Whereas if a gene arose way back in the last universal common ancestor of bacteria and archaea, you'd expect to find it relatively widely shared compared to a gene that's younger. There's many challenges with these kinds of methods, though, particularly for prokaryotes and across life. Also for eukaryotes, there's rampant gene loss. Prokaryotes have the particular problem of really rampant horizontal gene transfer. There's also some very difficult effects around length, that longer genes are easier to find homologs for. And it seems to be a robust result that older genes are nonetheless actually still longer on average. So when trying to classify proteins or genes or domains into age groups, you've got to be really careful regarding the length of the elements. Two main mitigating strategies that I've tried in this project so far is to really focus on the core genes that are relatively universal within a taxonomic group. So that they're, say, found in more than 50% of genomes or perhaps more. So the assumption being that accessory genes that are relatively rare are more likely to be those that are horizontally transferred, and so harder to assign a phylostratigraphic age to. And also, for various reasons, I've decided to focus on short genes and short domains because there's these are computationally a lot more easy to work with, and also because it removes some of the issues around the length effects that you've got to be careful of. Okay, so when thinking about how you might do this, probably the naive initial approach would just be see what's in AlphaFold. But the difficulty is that both AlphaFold and the ESM Atlas database, which contain hundreds of millions of proteins, so AlphaFold contains about 200 million proteins, ESM Atlas contains about 700 million proteins, but neither of these is taxonomically well balanced. So if you want kind of a broad analysis of the history of life, these are not going to be that useful, at least without taking some care around them. So as I was thinking through this, it seems that to me that probably some alternative to these is needed, or at least some very careful kind of processing of the data. Another tool that's very standard to use now in this kind of analysis is FoldSeq, which, to kind of simplify it, basically reduces down the complexity of a structural model into structural alphabet characters. So it reduces the structure down into an alphabet of 20 possible characters, which are called 3DI characters. And with these characters, that allows you to use kind of standard sequence search methods with this protein alphabet protein structure alphabet, rather than the normal alphabet of amino acids. So that's a nice method. It's very fast, which is great, and it allows you to search the whole AlphaFold database in just a few seconds or maybe a few minutes. But as I kind of looked into this further, I increasingly realized it has the really big disadvantage of it has a lot of false positive matches and also a vastly underestimated error rate, which means it's really not ideal for trying to do phylostratigraphy, because you really don't want false positive matches to distant, taxonomically distant proteins that are not actually related. And this seems to crop up again and again when you try and use something like FoldSeq. And I think it's basically just that there's not that much information in these 3DI characters, which means that false positives are quite easy to get. It's quite easy to get a similar sequence through chance rather than through evolution broadly. Okay, so what do we do? Do we try and compute a whole new structured database? Well, that's not really feasible. It's still too slow unless you have the massive resources of something like Meta, where you can use hundreds of GPUs. So while structure prediction is very fast with ESM-fold, on the order of seconds per protein on a good GPU, it's still too slow for millions of proteins to be feasible. There are a lot of recent developments in the space, but it's actually still a very messy field and quite difficult to actually get any of these new tools to work. And if you try them, they often don't produce structures that are very good, particularly when you're looking at young proteins, which are typically the proteins that don't yet have structures inferred for them. So I would say from so far, I'm curious what people think about this, but my sense is that if you really want massive scale structural inference, ESM-fold is probably about as good as you can do, but there are some new methods that people are developing. And I'm curious if someone has thoughts on that. Okay, so I think we ideally would have a protein structural database which is taxonomically unbiased, and I'm trying to kind of collate this from different sources by matching, I chose GTDB proteins to different structural databases. So firstly, AlphaFold, but then some of these other smaller databases that are specific to bacterial proteins, and also ESM-fold. And that allows you to catch to find a reasonably similar sequence for most of the sequences in the GTDB. GTDB is a taxonomically representative set of around 100,000 genomes across both bacteria and archaea. So this is still kind of in progress, trying to make the full database of this. We also need a more accurate search tool. I think the best one is ReSeq from Robert Edgar, which is a lot more accurate than FoldSeq, partly because it uses a much larger structural alphabet rather than just 20 characters. So yeah, if you're interested in looking into this area, I really encourage you to look into ReSeq. I think it is the best tool available at the moment. Okay, so we probably, so one of the underlying issues is we probably don't need to infer new structures for every sequence, given a lot of sequences, given one structure can represent a lot of sequences. But how do we optimize that choice between when we actually do need to calculate a new structure versus when we don't? The best I've come up with so far is to say if two sequences have the same secondary structure and clear homology at the sequence level, then I think it's reasonable that we don't need to calculate a new structure. This seems like a somewhat reasonable approach because it's a lot faster to infer secondary structure than it is to infer tertiary structure. There's other approaches. Maybe we could use these 3DI characters instead of secondary structure. We could go straight from protein language model embeddings. Again, curious for feedback in the Q&A, what people think is the best approach. Okay, so this is some very messy initial data. I'm sorry it's so it's so messy. But I think there really is a general trend that if we look at, so I've called this pseudo-age, so it's basically phylostrata. So the very youngest genes have tend to have a high proportion of alpha helices, like we saw before. The very oldest genes here that are shared across different phyla, either in bacteria or archaea, cluster with far fewer alpha helices and also more beta sheets. So this is very messy data, partly because of the issues with things like fold-seq that I'm working on. But the more I look into specific cases, I think there is this kind of general trend that the very oldest proteins tend to have acquired more beta sheets, and the very youngest proteins is quite rare. It's relatively rare for them to have beta sheets. Okay, so I think the specific results, I haven't seen that discussed before, the idea that young proteins tend to be more alpha helix rich. And I'm curious what people think about that. But then in general, whenever we see a long-term or a supposed long-term trend in protein evolution, we can ask what might be the driver of that. It's hard to imagine, and again, this is a point we can discuss, but it's hard to imagine natural selection as kind of a general explanation of very long-term trends. There's no expectation that natural selection would keep pushing protein evolution in a particular direction over literally billions of years. That would be very surprising on current theory. So I was thinking around some other options for what might explain even just the long-term trends that we've seen in the literature so far. Something that hasn't been developed that much in the context of protein evolution, but has been developed for RNA evolution, is called phenotypic bias. And this is this idea that some phenotypes are just much more common in sequence space than other phenotypes. So if you look at RNA secondary structures, there's only a certain small proportion of structures that are highly common in sequence space. If you look at natural structures, quite interestingly, they're basically all within this highly probable region. So the secondary structures of RNA that are found in nature, that we find in the RNA structure databases, correspond to a small subset of all the possible structures. And those structures are the ones that are somewhat biased in sequence space, if that makes sense. So if you randomly fold a RNA sequence, it tends to be one of just a few, it tends to fold into one of just a few structures, and it's those structures that we see in the natural data. And that's not what you'd expect if the selection, if the explanation of this is natural selection. If the explanation is, whatever's most optimal, you'd expect one of, any of these other kinds of structures could have evolved or could be quite common if it's natural selection that's choosing them. You might think, okay, it'll just be within that subset, the functional ones, but there's no particular reason to think that the functional RNA structures should correspond with those structures that are just highly probable because of the phenotypic space. So this is something that's been explored for RNA and is starting to be explored for protein. And I think it could be a potential explanation that, at least for new genes, I think this is quite clearly a good explanation that most random sequences are gonna fold into the most common structures. And so whenever you get a new gene, you'd expect it to be one of the structures that are relatively common in the sequence space. So, okay, what would that look like for protein evolution? Well, it turns out that alpha helices are quite a bit more common in sequence space than beta sheets. So if you randomly fold a sequence and predict it with basically any method you like, it's considerably more likely to fold into an alpha helix than it is into a beta sheet. In other words, there's a phenotypic bias in protein space towards helices. And I think that basically explains why the youngest proteins tend to be helical in their secondary structure. You can look at this various ways, but one of them is what happens if you just take non-coding open reading frames across diverse bacteria and see what's the proportion of helices to sheets. And in this case, I've just taken an arbitrary length, just 25 amino acids. And basically, there's always a 2-to-1 ratio or so of helices over sheets, sometimes more, depending on, well, I'm not sure exactly depending on what, but different genomes do have different intrinsic biases. It might be related to GC content, but I wasn't able to see a strong relationship there. So I think it's a bit more complicated than that. But yeah, if you take non-coding sequences in nature, they tend to be somewhat biased towards helices, and this is true of random sequences as well. Okay, so another thing that kind of builds into this idea that alpha helices are simpler than beta sheets is that it seems alpha helices evolve faster than beta sheets overall. It's not a massively clear distinction, but there is a higher rate of evolution. And this has been reported in various papers. I haven't actually cited them here, but if anyone's interested, I can find those references as well. But this is something that has been reported a few times in the literature, that typically residues in a helix will evolve faster than residues in a sheet. And thinking about this in terms of sequence space and phenotype space, I think helps to make sense of this, that helices are just simpler, they're more robust, more changes are permitted within a helix than are permitted within a sheet. In other words, helices are simpler, you could say. There's various kind of updates to this that would be good to explore. Okay, so that was kind of the initial work in my project. And then as I increasingly was thinking around sequence space, phenotype space, I thought it would be really nice to be able to work out how many sequences could fold to a given structure. There's some literature on this, and there's some quite recent developments around direct coupling analysis that actually allow you to estimate, if you basically give a for a given structure, how many sequences approximately could fold to that structure. And you see for something that's very simple, like just basically a single helix, the estimate is something like 1 in 10 to the 12 sequences would fold to that structure. Now, these numbers are probably not that reliable, but I think what's more interesting is the kind of relative numbers. So for a much more complex structure, there's far fewer sequences that will fold to that structure. And this also fits with broadly what you see in the literature. Again, I think the numbers are not anywhere near exact, but I think they're kind of the rough ordering of them, that even for structures with the same length of sequence, if it's, say, very tightly packed and a very complex structure with a lot of relationships between the residues, there's far fewer sequences will encode that structure. So this is the idea of sequence capacity, or relative sequence capacity, how many sequences would fold to a structure. And basically, and this is still work in progress, I see that the young proteins tend to look like this, and they tend to have a very large sequence capacity, and in other words, very many sequences will encode that structure, and the older domains tend to have a higher packing density and also a lower relative sequence capacity. So it really seems that young proteins are born simple, and insofar as they're foldable, it's because the kind of evolution has stumbled across structures that are quite easy to find in sequence space. It's not clear how quickly they gain complexity and how long these young proteins survive. Many of them won't survive very long. They won't last over many millions of years. They're probably not that useful, but occasionally they are. That seems to be the general story. What drives the shift to greater complexity? Do we need to invoke selection, or can we get the complexity for free in some sense? And this is the kind of idea that I'm exploring, that just by exploring approximately neutral networks of a structure, I think you can stumble across increased complexity in that structure. And so I also propose, maybe controversially, that the starting point, that because you necessarily start at the simplest possible structures, that basically implies that there is a directionality to the process, that because you've started at the simplest possible place, any movement forward will tend to be towards greater complexity. Yeah, and again, I'm curious what people think about that. So kind of this butchered quote that the arc of the protein universe is long, but maybe it bends towards complexity, or at least that's seems to be a real possibility. Okay, so that was the work in progress on long-term trends. And now some of the earlier work I've done, which provides some context for it, thinking about the origin of new genes, particularly from alternative reading frames. Alternative reading frames are probably most famously known in the case of overlapping genes, where you have two different ways to read the triplet sequence of a protein from the same set of nucleotides. This is an example from SARS-CoV-2, where these two proteins have both been studied relatively well. And one of the very rare cases where we actually have good protein structures for both possible functional proteins that are encoded by an overlapping gene pair. And this one's been studied relatively well compared to most overlapping genes. So overlapping genes are relatively well known about in the case of viruses, but they're actually also found in bacteria and diverse eukaryotes, also in archaea. Overlapping genes or alternative frame open reading frames are just one in a large set of possible kind of alternative ways to encode proteins to get translation from a genome, which is kind of outside what we'd consider the kind of canonical gene. So there's proteoforms where you have different versions, different lengths of a given proteins by having different start sites. There's dual coding RNA, so where an RNA has a function at the non-coding level, say a regulatory function, but it also actually encodes a small protein. You can occasionally get stop codon read-through, which extends the protein. Sometimes so-called pseudogenes are actually translated. And then there's many of these just short ORFs that are kind of missed by standard translation, standard annotation mechanisms or gene finders. So there's this huge area of underexplored translated sequences, and presumably this is where new genes come from in some sense. And my research over the last few years, a lot of it has been focused on this particular category of alt frame ORFs, or alternative frame proteins. So there's 6 possible reading frames for any given nucleotide sequence. Conceivably, any combination of those could encode a protein. Whether that protein's functional is obviously a separate question, but at least conceivably you can get translation in multiple frames. What does this mean for evolution? Well, it turns out there's actually multiple mechanisms which evolutionary mechanisms which can use this alternative frame sequence, so sequence out of the kind of canonical reading frame to encode a protein. There's the case I mentioned of overprinting or overlapping genes, where you get translation of both reading frames. But there's other ways which evolution can make use of alternative frame sequences. One is frame shift, mutations where the reading frame that is translated is shifted because of a genetic mutation. That means that the kind of default frame that's read is shifted by, say, 1 or 2 nucleotides. Conceivably, at least, you could also get inversion mutations where you get inversion within a gene, and then you actually, the ribosome actually read through what used to be the opposite, the antisense sequence, for instance. You can also get fusion of genes, and this fusion can be out of frame, which would mean that at least part of the alternative frame sequence would be read into the new protein. And you can also conceivably at least get stop codon read through, which reads into the next gene in a different frame. So there's many conceivable ways in which alternative frame sequence can be used in evolution. And there's kind of a growing literature on each of these cases showing examples where this has actually contributed to novel sequences within genes, or at least in the case of overprinting for completely new genes. This was from a couple of years ago that I wrote this, and the literature really has expanded since. I was particularly pleased to see a paper in Nature last year, which was exploring this idea of inversion mutations. I think this really hasn't been discussed much before at all, but it's possible to get microinversions within a gene, which means that you actually end up, and this is the diagram from my paper, just showing how you can end up translating some of the antisense sequence. And they looked at some specific examples and showed cases where it was either detrimental or beneficial, sometimes environmentally dependent, to have this kind of inversion mutation. Now, these alternative frame sequences have various interesting properties, partly because of the structure of the genetic code, and this is something I've written about a couple of places as well. One of the properties is that when you shift, if you shift the reading frame by just +1, you actually don't shift the hydrophobicity of the amino acid that's encoded as much as you would expect. Most people would assume a frame shift would be the worst thing you could do to a protein, other than just a direct truncation. But actually, in many cases, it is actually not that bad, because you tend to preserve something of the same hydrophobicity, even though you encode quite different amino acids. Also interesting is if you do translate the antisense of a sequence, if you think carefully about the structure of the genetic code, that basically means transitioning from a codon which has U in the second position to a codon that has A, which because of the way that the code is structured, is basically transitioning from a hydrophobic amino acid to a hydrophilic amino acid. And so there's this kind of direct opposite patterning in sense and antisense that's been discussed in various papers, and might explain the existence of some of these overlapping genes, why they can actually remarkably be useful genes, despite coming from what might seem at first to be random sequence. Is this actually useful for evolution? There's some cases where it has been shown it is, but as kind of a broad theory, there's a lot more work to do in making sense of how common it is, and whether it's really that important, or it's just a very occasional kind of effect that you see. Okay, so some concluding thoughts just about where I think the literature on protein evolution might be going. I think there's really exciting developments around protein language models that will mean it's really fast to compute structures. We're almost at the point where you can get the structure, the full kind of proteome of, say, a bacteria on a laptop within the order of an hour, if you have access to a GPU, or at least we're getting close to that, because it only takes on the order of 1 or 2 seconds per sequence, at least for the fastest predictors. I think we can really do a lot more by making use of the AlphaFold structures as templates for similar, when we come across a similar sequence that might have a slightly different fold. I think using templates would probably help to speed things up. Focusing on domains will also help to speed things up, because most of these methods are really length dependent. For a lot of things, secondary structure is actually sufficient, and it is a lot faster to infer. So there's a lot of opportunities for starting to use secondary structure, even if the tertiary structures aren't feasible. And like I said before, there's new methods that are using better structural alphabets, so for more precise searching than you can do with FoldSeq. It turns out that smaller protein language models can often get equivalent results, so that will help. That is helping to speed up various of these analyses. There's new developments in the software, the hardware as well. That all of this is kind of meaning that it's kind of faster and faster to do inference using protein language models. It's already, relatively cheap. It's feasible to fold tens of thousands of proteins just in a few hours of GPU time. With this, we can start to really understand this relationship between sequence and structure at kind of a vast scale that has been possible for RNA for quite a while, but hasn't been possible for protein structures. Still, a major caveat that the usefulness for orphan proteins is not so clear. Most of these methods seem to give much better results when there are homologs in existing data sets that are used for training. Okay, so that's the end of my talk. Very interested to keep working on these things, to talk with people who are working on these. I'm sure a lot of people in the audience know a lot about some of the things that I've touched on. So with that, I look forward to your questions.